Hello everyone, my name is Ajita Shree
My name is Krishna Pawan.
and we are from Dr. Hamim's lab from the Computer Science Department, IIT Kanpur. And today we are presenting our method,
Yes,
scDREAMER. The motivation behind our work is that there is a presence of complex scRNA dataset that are generated from different labs across different tissues, locations, and different laboratory conditions. And the data integration task is very important for comprehensive understanding of any biological system. And it also helps in the discovery of major and rare cell types. Along with that, the Atlas level integration improves the biological analysis across different species. But there are a lot of challenges in the data integration task as the data is generated from the heterogeneous sources and it has batch effects. So today we are presenting our method, which is for data integration of single cell data, which is a novel deep learning architecture with adversarial VAE and a batch classifier.
scDREAMER was tested against 5 real datasets, which are human pancreas, lung, immune human, macaque retina, and human mouse. Each dataset has its own challenges, namely, human pancreas is taken from different sequencing techniques, whereas lung has nested batch effects, and the macaque retina has a very large number of batches, more than 30, whereas immune human is taken from different tissue regions. Finally, the human mouse dataset consists of across species datasets, namely human and mouse. These 5 real datasets were compared against the best 6 state-of-the-art methods, namely SCVI, Harmony, Surrey, BBKNN, Scanorama, and Insight.
So here we explain the architecture of scDREAMER in detail. So the scDREAMER consists of adversarial VAE network and a batch classifier. So adversarial VAE network takes set of scRNA data from different batches as input and try to reconstruct the single cell data. Along with it, it learns the latent space representation of the entire data set. The discriminator network takes the scRNA data and the reconstructed matrix as input and try to classify between the two matrices. There's an adversarial training between discriminator and the autoencoder network. Batch classifier is introduced to improve the batch mixing within the dataset. And there we do the second set of adversarial training between batch classifier and autoencoder. So batch classifier learns cross-entropy laws and try to correctly classify the embeddings into the batches, whereas encoder is trying to generate the embeddings such that batch classifier is not able to correctly classify. The details on the loss function for both the training is on the left side of the slide.
To evaluate scDREAMER, we have introduced broadly 3 categories of metrics. One is bioconservation metrics, batch correction metrics, and finally the isolated label metrics. So for get an holistic view of bioconservation metrics, which involves NMI, ARI, and ASW. And similarly, for bioconservation metrics, we have batch, ASW, PCR batch, graph connectivity, and cable. On these two metrics, batch correction and bioconservation metrics, we have an combined composite score, which gets which has an holistic view, and the isolated label score, which was introduced for real cell types, has an isolated F1 score and an isolated Siloche width. On the other hand, we have other metrics such as graph CLC and the portion of positive cells and true positive cells.
Here we present the results on the human pancreas dataset. The dataset has batches from different sequencing protocol. There are total 9 batches in the data. And as we can see from panel A and B, scDREAMER is able to correctly classify different cell types into different clusters and attaining a very good batch mixing as shown in plot B. Quantitatively, we can see that our method is performing the best in both batch correction as well as bioconservation. In panel H, there is a large presence of positive cells denoted in green color, which shows that the separation between different cell types is very well attained. Whereas the blue cells, which are true positive cells, that indicates the batch mixing is very well attained.
Here we look into human mouse dataset, which was addressing the nested batch effects. It consists of donor level variability and sequencing protocol specific batch effects. The dataset consists of 10 batches, which are taken from 2 different tissue regions, namely bone marrow and PBMs. Quantitatively, if you look at the similar cell types of like CD4 plus and CD8 plus, which are also closely also closely present in the embedding space. If we quantitatively look at the results, like in the panel C, D, and E, we can see that scDREAMER was able to perform best in all the three metrics. Even in the bottom right, we can see that the very high percentage of true positive cells indicate that the cells were batches were really well integrated.
Here we present the results on the human mouse dataset. Here we are doing atlas level integration between two different species with about 1 million cells and 97 different cell types. As we can see from panel AB, that the batch mixing is very well attained between the species. And in the diagram below, we can see that there's a high percentage of true positive cells, which indicate a very good batch mixing. Quantitatively, we can see that in bioconservation, our method is performing the best and at par with the best method in the batch correction matrix. Overall, scDREAMER is performing very good on both the matrix. We also demonstrated scalability by comparing the runtime with other deep learning based methods, SCVI and Insight. And we can see from the graph below that our method has outperformed SCVI by 50% reduction in the total runtime.
So in the conclusion, firstly, we can say that scDREAMER was able to achieve 5 to 20% of improvement over the existing benchmarking methods, which was demonstrated on 5 real data sets. Secondly, the scalability to million cells and the performance of atlas level integration was also demonstrated in human mouse dataset. And we can say that scDREAMER was able to have best batch correction while maintaining its bioconservation metrics. Thank you.
Thank you.